Thank you all for joining us for our live webinar, which will be presented live by our guest speaker, Yutin Wong, entitled Lipidomics in Rare Disease Diagnosis and Therapeutic Monitoring. My name's Bill Griffiths, and I'm currently based in Swansea in the Oxysterol Research Group. The webinar will be around 45 minutes, allowing 10 minutes at the end for questions. Now I'd like to introduce our speaker. Professor Yutin Wong is a professor of biochemistry. She obtained her BSc in pharmacy from Beijing Medical University and a PhD in biochemistry at Karolinska Institute in Stockholm. After a PhD, she moved into proteomics, first in the biotech industry and then at University College London. In 2007, she saw the light and moved into lipidomics. She has extensive experience in applying mass spectrometry-based lipidomics to identifying aberrant cholesterol metabolites associated with dysregulated cholesterol metabolism and in studying sterol-protein interactions. Okay, Yutin, the floor is yours.
Thank you very much, Bill, for the introduction. So first, let me share my slides. Okay, thank you very much. Hello, I'm Yutin Wang. So today I'm going to present some recent work we have done as part of Rare Disease Research Platform UK, which was launched in July 2023 with £14 million investment from UK Medical Research Council and also National Institute for Health and Care Research. The platform is made up of 11 research centers, also known as nodes. One of the nodes is rare disease, lipidomics, and metabolomics for rare disease diagnosis. In Europe, a rare disease is officially defined as life-threatening or chronically debilitating condition that affects less than 1 in 2,000 people. There are estimated over 7,000 rare diseases. Although individually rare, collectively they affect between 27 to 36 million people across EU. 80% of rare diseases have identified genetic origins. There are many challenges in rare disease. So one is diagnosis delay. So average is patient experience 4 to 5 years diagnosis delay. And 70% of rare disease affect children. 3 out of 10 of these children do not survive past the age of 5. And only 5% of the disease have an approved treatment. There is still much work to be done to improve the diagnosis and the treatment of rare disease. Recent advancements in genomic sequence revolutionized rare disease diagnosis. So as example, the 100,000 Genome Project, which is Genomics England, a very first initiative. They sequence 100,000 genomes from patients affected by either rare disease or cancer. In this project, 190 rare diseases were included and people invited to take part were thought to have one of these conditions, but not have a molecular diagnosis. At the end of this project, 25% of the patients received actionable funding. However, 75% of the patients still remain without a clear diagnosis. So this demonstrates both the promise and the limitation of genome sequence for rare disease diagnosis. Even with a full genome is available, it remains difficult to interpret. A genome sequence contains millions of variants compared to the reference genome. The challenge is identifying which variant is actually disease pathogenic. So many variants are classified as variants of uncertain significance. Therefore, different type of functional assay validates the variants, and multi-omic approach is required. A large subfamily of rare disease are inherited metabolic disorders, IMD. These include disorders of amino acid metabolism, carbohydrate metabolism, lipid metabolism, or cofactors and vitamins. So these disorders are usually caused by genetic mutations that affect enzymes or transporters, or organelle proteins dysfunction, such as mitochondria or peroxisome protein dysfunction. Currently in the UK, only 6 of IMD are included in the newborn screening. But then the total number of different IMDs actually is above 1,500. So in today's talk, I will focus on the disorder of lipid metabolism. If a patient presents with non-specific or unexpected symptoms, initially doctor will request a general test. If the basic investigation indicates or suggests it's an IMD, specific tests will be required to perform in the hospital metabolic lab. And normally now in the parallel, a whole genome sequence will also be requested. So for inborn error of metabolic disorder, mass spectrometry has long been used to help to diagnose the disease. So the first case study is a fatty acid metabolism disorder. Reflux disease is a peroxisome disorder that due to deficiency in the phenyl-CoA hydroxylase, which normally catalyzes the alpha oxidation of branched-chain fatty acids in peroxisome. In this case study, there are two siblings. They have displayed a clinical symptom, which is suspicion of reflux disease. Genetic testing confirmed these two siblings had the identical mutation in PHPY gene. However, very interestingly, only one of the siblings displayed the skeletal abnormalities. Therefore, their plasma sample was sent to our lab to perform a shotgun lipidomics to analyze their lipid profile. So, just as shown in the bottom, normally this enzyme PHPY will metabolize the phthalic acid, which is a carbon-20 zero branched chain fatty acid. In human, we obtained from our diet. They can come from either dairy products or ruminant animal fat, and also some certain fish. Briefly, for the shotgun lipidomics, we extract 5 to 10 microliter plasma and then add the internal standard splash lipidomics. We perform the shotgun lipidomic analysis using nanomeds with IQX Orbitrap. The high-resolution mass spectrometry was recorded in Orbitrap, while MS fragmentation was also performed. Then the data was searched against Lipid Explorer software. As shown in the bottom row, the first figure is the ratio between the carbon-20 zero fatty acids to carbon-19 zero fatty acids. As expected, the abundance of ratio of carbon-20 zero to carbon-19 zero fatty acids was elevated compared to the control. Here we use the NIST plasma. So in both patients, it's around maybe between 4 to 5 times higher, the ratio compared to the NIST. And then we also observed the carbon-20 zero fatty acid content in cholesterol ester is both elevated in patient 1 and patient 2. So as the complex lipids, like for example diethylglycerol and phosphatidylglycerol. So between these two patients, they have displayed a very similar lipid profile, except the one I listed here, we didn't see any other changes. So however, it's the case 1, the patient 1, which has the skeletal abnormalities. But then the lipid profile between case 1 and case 2 are very similar. Or you could say that in case 2, the carbon-20 zero fatty acid actually is slightly higher than compared to patient 1. So this is an interesting example. The diagnosis is straightforward. However, the genetic information and the lipid profile didn't explain the observed difference in the clinical manifestation in these two siblings. So it suggests other factors such as epigenetic regulation may play a role. So this requires further study to dissect which mechanisms cause a specific symptom. Next, I'm going to change the topic from fatty acid metabolism to cholesterol metabolism. During cholesterol metabolism, the first step is to add a hydroxyl group to the molecule, which are catalyzed by different enzymes. The expression of these enzymes, such as CYP7A1, CYP46A1, CH25H, or CYP26A1, are cell type and tissue specific. Like CYP7A1 mainly expressed in liver and CYP46A1 expressed in neuron. CH25H in normal condition expressed very low in the immune cells, such as macrophages, and CYP27A1 expressed ubiquitously. The challenge in study cholesterol metabolism using mass spec is first the metabolite of the cholesterol. Although cholesterol is very abundant in the blood, there is metabolite normally present at least 1,000 times less compared to cholesterol. Also look at this molecule. There's no charged group for either positive or negative analysis in the electrospray mass spec. So we have developed a method called enzyme acid dimerization for stereo analysis. So in this method, we will first use cholesterol oxidase, convert the hydroxyl group to a keto group. Then that can easily react with the hydrogen. In this dimerization, we add a positively charged nitrogen onto the molecule. After dimerization, this will enhance the signal in mass spec analysis at least 100 times more. The other advantage is after dimerization, it directs the fragmentation, which gives a characterizing fragmentation pattern. So we normally perform our analysis use the hybrid ion trap and orbitrap instruments. So the spectrum shown here is the MS3 fragmentation spectrum. This is listed as 4 different isomers of monohydroxycholesterol, such as 24S hydroxycholesterol, 25 hydroxycholesterol, 26 hydroxycholesterol, normally people call it 27 hydroxycholesterol, and also 7-alpha hydroxycholesterol. So in the fragmentation, after dimerization, they first lose the pyridine group in the MS2, and then we can take the MS2 abundant ions to a MS3 fragmentation. So if you look at this MS3 spectrum, although the fragments are more or less similar in all these isomers, however, their patterns are very different. So for example, between the 24S to the 26 hydroxycholesterol, the biggest peak here, M over Z peak, 437, is high in 24S hydroxycholesterol, but it's lower in 26 hydroxycholesterol. So in this way, in our analysis, we can identify the different oxy-zero and quantify it based on the accurate mass and also the MS3 fragmentation, which compared or matched to the authentic standards. So this is just an example of the reconstructed chromatogram of monohydroxycholesterol from the NIST reference plasma. With this method, we call it stereomics. So it basically can analyze from monohydroxycholesterol down to cholesterolic acid. So this is a stereomics analysis from 100 human healthy control plasma. This shows the profile, the variation in these 100 control donors. So their level varies between like low nanogram per ml in plasma, such as the 7-alpha 25 dihydroxycholesterol to like cholesterolic acid, which is above 100 nanogram per ml. So the second case study I will show is due to the deficiency of sterol 27 hydroxylase, CYP27A1. So the disorder is called CTX. So in this case, an adult who displays CTX-like symptoms, and genetic testing shows one allele of the CYP27A1 gene has pathogenic variant, while the other allele appeared normal. So the biochemical test also done in the hospital clinical chemistry lab. As shown here, the CYP27A1 is an enzyme which adds first a hydroxy group at the 26th position. Then the same enzyme converts the hydroxy group into a carboxylic acid group. So CYP27A1 is a key enzyme in bile acid biosynthesis. There are two major pathways for bile acid biosynthesis. On the right-hand side is the acidic pathway. The first enzyme is CYP27A1 in this pathway. As I just mentioned, so it's CYP27A1 will convert cholesterol to cholesterolic acid. So if you have a deficiency of this enzyme, so the patient will have this metabolites, this 3-beta hydrocholesterol acid will diminish in the patient's plasma. And this enzyme CYP27A1 also works in the classic pathway, the neutral pathway. So it is at this step converts the 7-alpha-12-alpha cholestanol to the cholestanoic acid. So, however, in the clinical biochemistry lab, they won't analyze any of these metabolites. What they will do, they either measure this cholestanol in the plasma, or measure the bioalcohol glucuronide in plasma or urine. They can just measure this metabolites this in negative mode, which is easy to do. However, in these patients, normally in classical CTX patient, this metabolite is significantly elevated. However, they only detect a trace amount of the bioalcohol glucuronide in this patient's urine. Again, this sample was sent to our lab for stereomic analysis. Using our EFSA method, we first checked the cholestanoic acid level in these patients. The top panel is the NIST reference material. This metabolite normally is abundant in plasma over 100 nanogram per ml. And then in the patients, the intensity of this peak is reduced. It's around 10% of the normal peak. You could say maybe this patient has 10% residue activity of this enzyme. Then we also looked at the 7-alpha-12-alpha dihydroxycholestanol, which is highly elevated in the patient. But then normally in the normal people, this metabolite in the plasma is very low. So it's fastly converted to the bile acid. However, in this patient, you can see it's accumulates due to the reduced activity of CYP27A1. If we calculate the ratio, use the 7-alpha-12-alpha dihydroxycholestanol divided by 3-beta-HCA. So the ratio of these patients definitely classified the patient as a classical CTX. Although at the moment, genetic only found one allele has the pathogenic variant, and then in the clinical biochemistry lab didn't see the bioalcohol glucuronide go up. So based on our analysis, we feedback for the genetics to reanalyze the data to check the other allele. At the same time, we have collected the sample from the patient's parents who will be the carrier of CTX to confirm if this patient is a classic CTX or if it's because it's a heterozygote. Normally, the CTX disease is autosomal recessive, and then people very seldom analyze either heterozygote carrier. So there's no any data to compare. So we hopefully by analyze these patients, the parents' plasma sample can confirm whether this patient is a classical CTX. For cholesterol metabolites, they also enzyme involved or protein involved in the cholesterol transport. Normally, cholesterol we obtained from the diet or synthesized in liver, which will be packed in lipoprotein particle as cholesterol ester. So this lipoprotein particle will circulate in the blood, and cells in the different tissue, for example, the macrophages, uptake the LDL particles through the LDL receptor. The LDL particle will enter the lysosome. In lysosome, there are enzymes called lysosome acid lipase, which will hydrolyze either cholesterol ester or triglycerides to release free fatty acids and cholesterol. Then cholesterol will be transported out of lysosome by the transport proteins such as MPC2 and MPC1. Cholesterol will be transported to plasma membrane or to ER for the cell to reuse for the cell divide and growth. If there are any deficiency in either the acids, lysosome acid lipase, which called LIPA, or the Niemann-Pick protein, which the cholesterol transport, which will result in the lysosome storage disorder, such as Niemann-Pick type C disease and Wolman disease. When the cholesterol ester is accumulated to lysosome, it causes oxidative stress, and then because there's no free cholesterol is released from a lysosome, the cell itself turn on their endogenous cholesterol biosynthesis. With the generated cholesterol react with reactive oxygen species, and then they generate all these non-enzymatic formed oxysterols in vivo. So actually, in both the cholesterol-related lysosome storage disorder like the Niemann-Pick type C disease or the Wolman disease, in the in the patient's blood, they have this characteristic non-enzymatic form oxysterols. Normally, this reactive oxygen reacts at the 6th position. For example, this is 5-alpha 6-beta, 3-beta 5-alpha 6-beta trial. Normally, we call it C trial. And then this form in vivo, then they can be converted by the enzyme in vivo, like CYP27A1, become a C trial acid. They also can form the 7-beta oxidized cholesterol or the 7-keto oxidized cholesterol. So you can use this in vivo formed oxidized cholesterol as the biomarker to identify the lysosome storage disease like NPC and woman disease. So woman disease is due to the deficiency of the lysosome acid lipase. So there is the range, it depends on the residue activity. Woman is a severe form of this lysosome lysase deficiency. So basically, it's completely lost the enzyme activity. So woman disease, no residue activity is present, leading to severe disorder that present during infancy. So these infants have a rapidly devastating cause of the disease. The deaths occur, without treatment, the deaths occur in the first year of life. So luckily, there are different options to treat a woman disease now. First is the FDA-approved enzyme replacement therapy. So with enzyme replacement therapy, the endogenous enzyme will be picked up by the deficient cells, such like macrophages. Then the enzyme will, through the endocytosis, enter the lysosome. Then in the lysosome, the functional enzyme will catalyze the hydrolysis of cholesterol ester or the triglycerides. So normally the enzyme replacement therapy will be given together with the strict diet to reduce or completely fat-free diet. However, with enzyme replacement therapy, the response in the patients varies and the treatment can be complicated. So the people also developed another therapy is hematopoietic stem cell transplantation. So just to go through this stem cell transplantation, first you will find a matched donor. The donor will donate the hematopoietic stem cells, and then normally the recipients will go through chemotherapy conditioning. This is to use chemotherapy to ablate the host bone marrow cells to create the space for the donor's bone marrow cells, and sometimes to suppress the host immune system in case they reject the donor's cells. Then the transplantation can go ahead, and then hopefully the donor cells will engraft in the bone marrow. Then the final step will be metabolic correction. So it just shown here, the macrophages will through endocytosis take the enzyme secreted by the healthy donor. There, the enzyme in the lysosome will function to release the cholesterol, to hydrolyze cholesterol esters. Through collaboration with clinicians at Manchester, we have been participates to use the stereomics method to monitor the response after the stem cell transplantation. So in this graph, what we have measured is, as what I mentioned before, is this non-enzymatic oxidized cholesterol, like 7-keto cholesterol, 6-beta or 7-beta hydroxy cholesterol. So the first is the before the transplantation. The middle point is after transplantation. On the last, this is the level in the NIST reference material. So here it demonstrates after the stem cell transplantation, this non-enzyme auto-oxidized oxysterol reached to the normal level. That shows the transplantation works, and then the metabolic correction worked in this patient. So through the collaboration with the Manchester clinicians, we found a very interesting, a new type of woman disease. Woman disease, in these patients, this actually a group of patients unrelated, they have a deletion of the LIPA. The genomics sequence analysis shows the deletion is not only in the LIPA gene, adjacent to LIPA is the CH25H. Also, CH25H is completely deleted in these patients. CH25H is an enzyme normally is expressed very low is only under like infectious condition, for example, such as virus infection or bacterial infection. CH25H is hugely induced in macrophages. They produce 25 hydroxy cholesterol. So this cholesterol has the antiviral and antibacterial activity. And 25 hydroxy cholesterol can be continued metabolized by CYP7B1, which form the 7-alpha-25 dihydroxy cholesterol. This oxysterol is a ligand for the EBI2, which directs B cell migration. So this is the first ever human patient we found has the CH25H completely deletion. Again, we monitor the stem cell transplantation treatment in this patient. So in this example, we have done a time course is before the treatment, and then here shows the months after treatment. We also analyze the patient's parents, the mother and father, which will be the carrier. So here, if we look at 25 hydroxy cholesterol, if we focus on the patient 1, so before the transplantation, 25 hydroxy cholesterol is completely non-detectable, is zero because the deletion of CH25H. So after transplantation, this is a half month after transplantation, 25 is high, and then after 1 month it's go down, and then we have monitored this patient up to 3.5 months, is kept at the normal range compared to the parents. And the second patient we have monitored up to 40 months is also kept at the normal range. It demonstrates the successful stem cell transplantation. And in these patients, we also monitor this auto-oxidized cholesterol metabolites like the C-trial and then the C-trial acid before and post stem cell transplantation, which is to demonstrate if the lysosome acid lipase has also been functioning in this patient. So if we look at the middle one, so at the beginning is before transplantation is very high, and then this around maybe 4 months after transplantation didn't have a big difference. However, after 4 months, this auto-oxidized cholesterol metabolites start to drop down. Hasn't reached the normal range, but we will continue monitor these patients. I hope now with these 3 cases I show, one is about fatty acids metabolism disorder, 2 is in cholesterol metabolism disorder. So what we show here with lipidomics, what we can do well is to characterize the variant of uncertain significance, and also we can use lipidomics to monitor disease progression and response to therapy. So what I didn't show here is really a big challenge. We also, in this project, we have analyzed sample from the patient. Is syndrome without a name. It's called SWAN patient. So with SWAN patient, at the moment, neither genetics, first genetics couldn't really indicate any gene has any pathogenic variants. And when we perform the lipidomic analysis, we have some very interesting data which shows the lipid change. However, because in the adult, most of these patients, the SWAN patients are adults, when we identify some lipid change, at the moment the challenge is to really to distinguish if this is due to the diet or other metabolic associated syndrome in this adult patient, or is due to any particular gene, or if it's just a secondary effect of a particular gene has mutation. So that remain a big challenge with the SWAN patients. So here I listed is what we have done with different disease. For example, to diagnose the variant of uncertain significance, we have done like Niemann-Pick disease, type C disease in adults, and SPG5, that is due to mutation in CYP7B1. So CTX, talks that's due to deficiency of CYP27A1. We use the lipidomics approach to monitor the response to therapy in the woman disease. And then we also study some other lipid-related disease just to study the biochemistry change to hope to understand the pathogenic mechanisms to support the development of the therapeutics. And then finally, I would like to acknowledge all the collaborators for the study I present here. So this node, lipidomics and metabolomics for rare disease diagnosis node, is a collaboration between different sites in UK. from Swansea to Cardiff, and UCL, and also Manchester and Sheffield. And finally, thank you very much for your attention. I'm happy to take any questions.
Okay, thanks, Yutin. That was a really interesting talk. So our first question is from Alicia.
Very nice talk. Thank you very much. This is a really great job and really needed. And I was wondering, you mentioned, I'm not sure I get it right, that you are measuring bile acid and oxysterols with shotgun lipidomics.
Yes.
So, and you are doing a derivatization to see this kind of molecules. I was wondering if you also try without derivatization to see them, because I think they are really difficult to be detected in this way. So I wanted some comment from you about the
technique. So in the lab, me and Bill, we always use derivatization, but I know there are like other group, like Jeff McDonald's in US, he just used the triple core to monitor the MRM without derivatization. I guess it definitely works well in his hand. So it works, and then this method, different method has different advantage. We prefer the derivatization because this gives us the specific fragmentation. But then without derivatization is much quicker. The sample preparation is easier.
Okay, yes, I was thinking about that. Thank you very much.
The next question is from Jen Phillip.
Yes.
Yeah, that was really lovely. I just had one question, which was, I noticed in the patients who had the 25 hydroxylase deficiency, they had like unmeasurable 25-HC, but then they had some other metabolites, like I think 725 and some others, where there was the 25 hydroxylation.
Yes.
So I was just wondering how that, like where you think that's coming from or how that's happening.
Well, Jay, you are really spot on. That's correct. Although they don't produce 25, we did detect 7-alpha 25 dihydroxycholesterol. I believe that is first it had the 7-alpha hydroxylation, and then they add through a completely different enzyme, not CH25H. At the 25 hydroxy group on. So I'm not so sure which enzyme do it. Maybe Bill.
I think it's probably CYP3A4.
But very interesting. Yes, in this patient, we still see the 7-alpha 25.
I also noticed that like IFIT2 is in the same region of the genome, which is just interesting since that's also an interferon inducible gene going the other direction. But could imagine that like epigenetic regulation links the expression of the CH25 and the IFIT2.
Okay.
I mean, since they're both interferon regulated, that's just like a prominent interferon regulated gene that was right next door.
Okay, thanks, Jen. The next question comes from Joseph. Okay, this is Joseph. Thanks for the talk. Thinking about the Refsum disease work, has there been similar exploration in X-linked ALD and other peroxisomal disorders?
We did run a few because we did lipidomics and plus stereomics. That's what we routinely do in our lab. I guess we didn't see anything more than what is expected.
Okay, I will ask you a question, Yutin. So, you use this derivatization. What's the most challenging part of that derivatization, and why do you think perhaps it doesn't become more popular?
I guess it's the time. It takes 3 days, but then the derivatization itself is very easy to do. It's just the time. That's my opinion.
Okay, then as a follow-on, so at the moment it's sort of done in a university lab. How easy do you think this would go to move it from a university lab to a healthcare setting, like in the UK, into the NHS? Is that practical in the foreseeable future?
Well, I think it is feasible because if you have the method, everything in a kit format, all the region is included in the kit, and then at the moment we do the derivatization overnight, but in reality it doesn't need that long. 2 hours is enough to study this inborn errors of cholesterol metabolism disorder is no problem because the change is huge. So I
Okay.
think it's possible.
And besides you picked on CTX and Wolman's, are there many other diseases that the method's particularly good for?
I will think it's SPG5, which at the moment is not routinely analyzed by the clinical chemistry lab. So that is the need for the method to set up in the clinical chemistry lab.
Okay. I'll wind up the session. Thank you very much, Yutin, for the very interesting seminar. And also thanks for everybody for coming to listen.
Thanks.
Bye-bye.